Now we're gonna see what are the inhibitors of translation. So mainly we're gonna concentrate on large group of antibiotics like aminoglycosides. So where streptomycin, kanamycin, tobramycin all come under this group. So large group of antibiotics and how they gonna hinder the translation process, or on what step or on what process the translation is getting hindered so that there is no protein produced in our body. So we're gonna see that process in detail. So we're gonna see inhibitors of translation. So under inhibitors of translation, the first antibiotic we're gonna see is streptomycin. So it is the first, if you have first certain points, it is the first antibiotic aminoglycosides. And the second point, it was discovered in the year 1943. The mode of action of streptomycin is, how it is gonna inhibit the translation is, it is gonna bind to the 60S rRNA. So it is nothing but ribosomal RNA. And this process is being carried out in such a way that your P site, the P site of the ribosome can no longer bind to the chemically modified charged tRNA. The first antibiotic is the streptomycin. So you all know in translation process it is divided into initiation, elongation, termination. You have a role of two ribosomes, that is a smaller subunit 30S ribosome and a larger subunit 50S ribosome. So you have a site, three sites known as E site, P site and A site. So E is nothing but HCG site, peptidyl formation site, and A site is amino acid transferase addition site. So in P site is the first molecule which is free, or the site which is free, where chemically modified, for example F-methionine, formulated methionine comes and binds with a charged tRNA, comes and binds in the P site. So this is the process of initiation of a translation. So the streptomycin hinders in this process itself. So the no longer F-met will come and bind to the P site. So this is the role of streptomycin and how it is hindering the translation. The second one is the gentamicin. Streptomycin. The second one is the gentamicin. So it is also comes under aminoglycoside antibiotic. The mode of action of how it is been acting is, so gentamicin is a aminoglycoside antibiotic, and the third one, I'll tell you the mode of action together for two, three antibiotics which have a similar mode of action. So gentamicin, kanamycin, and the other one is the tobramycin. So these antibiotics are aminoglycoside antibiotic. The mode of action is, it is gonna bind to the 30S subunit of your ribosome. So if it is getting bound to the 30S subunit of ribosome, it can no longer bind to the 50S subunit. So the 30U subunit cannot bind to 50S subunit. So what happens? The translation gets hindered. Only when 30 and 50S subunits are bound together, the translation process of elongation takes place. So if it is not binding together, then the process gets hindered in the initiation itself. So 30S, and it is gonna bind to 30S and it is gonna hinder the addition or the binding of 50S subunit to the 30S subunit. The fifth one is tetracycline. So it is a group of antibiotic. It does not come under aminoglycoside antibiotics, tetracycline, and it is the mode of action is blocking the binding of charged tRNA molecules, that is new amino acid carrying tRNA molecule, to A site. So you know what is A site? It is amino acid tRNA addition site. If this is blocked, no mer. So first formulator methionine comes and binds to the P site. For the next amino acid carrier to be bound, it will be bound to the A site. That's the amino acid tRNA addition site. If it is getting blocked, then no longer this process is continuing. So the translation gets hindered. So no protein will be produced. Next is the macrolides. So these macrolides are characterized by the lactone rings linked to glycoside bonds. Macrolide antibiotics, lactone rings they will have, and it is linked with glycoside bonds. The mode of action is, it is binding to 30S subunit. It has so many of action. So binding to 30S subunit. It also prevents elongation by inhibiting peptide transferase activity. So macrolides will have a glycosidic linkages with the lactone ring. This is a large group of antibiotic also, and this antibiotic is gonna bind to the 30S subunit. If it is bind to 30S subunit, no way the smaller subunit cannot bind to the larger subunit, that is 50S subunit. The second one is inhibiting the role of peptide transferase activity. So what is peptide transferase activity? You all know that peptide bond will be supplied by the peptide transferase enzyme. Only 50S subunit can do that. So when the peptide bond formation is hindered, there is no long chain of peptides will be there. So no protein will be produced. So this process is being hindered by macrolides. Chloroamphenicol. This also hinders the peptide transferase activity. And it is selective only for 70S ribosome, and it is not seen to affect 80S ribosome. So this have to be known, not seen to affect the 80S ribosome. So you have inhibitors of translation. There are various antibiotics which is going to inhibit the translation. So one or the other way, either in the initiation or in the elongation process. So if the process is hindered, there is no protein production. So we have seen a large variety of aminoglycoside antibiotics like streptomycin, which is going to bind to 16S rRNA, which is going to chemically modified FMET will come and attach to the P site, it is hindered. Next is gentamicin, kanamycin and tobramycin. They are also aminoglycoside antibiotic. It is going to bind to 30S subunit, so that 50S subunit cannot come and bind to 30S subunit. Next is a tetracycline. It is a large variety of an antibiotic. It is going to block the charged tRNA binding to the A site. So A site is nothing but amino acid tRNA addition site. So if no new amino acid is been carried by the charged tRNA, then the process is getting hindered, because only one amino acid is already there. The new amino acids are not getting in. Next group of antibiotic is macrolide with the lactone and a glycosidic linkage to be specific, and it is also binding to 30S subunit, and it is going to hinder the function of peptide transferase by bond formation. So if peptide transferase activity is hindered, no peptide bond formation will be there. Without the peptide bond, the link will not be complete. So there is protein inhibition. Now chloroamphenicol, it is also have the same role, that is inhibiting the peptide transferase activity formation.